Hi everyone, and welcome to ISCT TV 2026. I'm Patrick Hanley, and your co-host for ISCT TV, and it's a pleasure to be here with Alicia from Down Under in New Zealand. Welcome, Alicia.
Thank you, Patrick.
Yeah. So today we're going to talk about what we've seen on Thursday, and maybe I'll start with you.
Okay,
what is the takeaway?
It's been pretty buzzy today, actually. There's so much going on and there's so much energy, which has been fantastic. I've seen some amazing abstract presentations. I've really been enjoying the science today. There's some outstanding stuff. Saw a couple of Kiwis and a couple of Australians doing some great presentations, so that's always good to see. And is one of the roundtables, which was actually chaired by our very own Mikey, which was fantastic. Talking about this cross-border access and all of the complications that roll on from trying to do something like that, even within a cohesive system like the EU. And I'm always looking at these things with a perspective for our region, but it's just interesting to see the crazy level of complexity once you try and do something, not just within different hospitals, but actually across different jurisdictions, and it gets really, really tricky. But one of the panelists, they were fantastic panelists, but one of them had this amazing sort of way of boiling it down, and she was like, there's three things that you need to be thinking about. It's the product flow, the patient flow, and the money flow. And sort of the take home from that is phenomenal, because I think we can apply that to all of the things that we're doing, where all of those things have to align to make it work well. They're just absolutely amazing sessions.
Yeah.
How about you?
So there's three things I want to touch on, but I also want to start with reflecting on our citations and champagne meeting this morning.
Was there champagne?
There was not champagne.
Oh, what?
I don't know if it was a regulation. I don't know why. Ed didn't tell us, but we had orange juice.
Oh, I'm sorry.
It's
At 7 a.m.
At 7 a.m. So we'll work on that for next year. I don't know if the Canadians will let us drink at 7, but we'll see. So the three things that I heard, there was a lot of buzz around access, and you and I had talked about this before, where in the past access meant decentralized manufacturing or these other things. For me, what I'm hearing is, no, how do we bring this into the community? How do we make sure that the other 80% of patients are getting access to it? So it's not as much about where it's manufactured, but even the commercial products, how do we make sure that those are for everyone and not just wealthy people that can pay for them and things like that? So that's one of the things. The second is the different cell types, right? So we work on I feel like a pretty broad range of cells at Children's National. So mesenchymal stromal cells, CAR T cells, virus T cells, tumor antigen-specific T cells.
Yep.
Just had a paper accepted in Nature Medicine yesterday.
That's so exciting.
Thank you. But anyways, I'm learning about macrophages. I went to 2 talks today that were about macrophages. So in one case...
I love this.
No!
Part of my PhD was in macrophages.
Oh, all right. I was wondering why you're getting so excited. I don't know anything about macrophages, but no, they could polarize these macrophages and use them in liver disease, such as cirrhosis.
Yeah.
And so that's exciting, not even gene modified. I just came from an oral abstract session for immunotherapy, learning about T regulatory cells, which I feel have flown under the radar for a very long time, and they've kind of reemerged.
Yeah,
so
definitely.
We'll see about those. And then there was another macrophage session actually using biomaterials for fibrosis.
Oh, I saw that. That was fantastic.
Yep.
Yeah,
so
that was really exciting.
Up to the last minute, I was at the oral presentations.
There's so much going on, right? Like, there's so much happening, and there's such a huge buzz and groundswell of, like, here we are, we're all together, and we're having this opportunity to talk and to catch up on the new stuff and to reflect on the stuff that's progressing and what's not and what can we do better. And it's one of my favorite things about this meeting is those opportunities to connect and see all the different bits at once.
Yep, agreed. So what about for Friday? What's in store?
Okay, well, I'm going to do a plug because we're holding the Mentoring Summit. Very, very important for our ESP community, but also for our more established members. So fantastic opportunity to, give a little bit of advice, come and share that, and it's really been a critical part of our ESP experience and community. So
So as
the mentoring summit.
but as the ESP superstar, I'm sorry if I'm changing the name of the word, but can you talk more about where that program stands? When I was involved in founding it in 2015, I think there was one committee of maybe 12 people. I don't know the exact number. But from my understanding, there's now regional ESP committees. I think there's 100 mentees or something. I don't know what the numbers are, but
it's exploded. It's exploded. So we have our global ESP committee, which is very large, and then we're split into sub working groups, and one of the working groups is a mentoring committee. And last year, I think we had close to 200 mentees. So it's really well established now. And then what's been happening is that ANZ, of course, basically established a regional subcommittee for ESPs, and they've been very active in their region. And now we're seeing actually potentially, I don't know if I can talk about this even, there's going to be an upcoming EU committee as well, which is going to be really exciting.
Great. I love seeing how much it's grown. It's so rewarding for me to see. And you know, it's why it's important to have turnover. You know, I would have never envisioned having all these different committees and things. Like, we just wanted to get it started, maybe have some presence. I think that was the new thing, the new initiative that we started, was trying to place an ESP in every scientific committee, and that was the extent of it, right?
And that's been amazing. And you see, again, the development of leadership as people are involved in the committees, and then they grow into actual, you know, other committees, and it's just fantastic. It's a really good way of doing it. It's interesting. I think there's a lot of need driving it, because, and I was saying to someone the other day, this is still a new developing field. There aren't any clear pathways for us, but there's a lot of people who want to get involved, and how do you do that? And this is a fantastic opportunity for that. It
was also interesting to see that there's more attendees from Europe than in the United States. And I think that's important for the society as it grows. It can't just be the United States. It needs to be a global society, right? And so that's really encouraging to me.
Definitely.
All right, well, I think that's a wrap for Thursday. I want to end by encouraging everyone to reach out to us, Alicia and I. One of my favorite parts of ISCT is networking. So let's see how many of you we can connect with after the annual meeting.
Absolutely. Looking forward to tomorrow.
Yep.
Hello from Dublin. I'm here at ISCT 2026, and I'm here with Bernd Giebel, and we are going to discuss EV therapies today. Hi, Bernd. How are you?
Hi, Eleni. Great. So happy to sit with you here on the couch.
Yeah, thank you for being here. So, you know, EVs have been a buzzword for a few years now. However, we have not seen any approved products. What do you think is some of the main reasons for that?
So it's not that long. So if we think about the first report about application, it was in 2014 reported. And if you compare it with the MSc field, so 1999 they started, first application was 2004. And now how many approved products do we have? I think worldwide 13, with the first in the United States 2024. So that means it takes time, and yes, we have a couple of challenges to solve.
So what are some of the regulatory challenges that you think the biggest ones?
I think the regulators, for them it's new, but at least in Germany we discussed with them. It was not so difficult. We need to discuss with them, certainly. But what I experienced in the last few years is the largest challenge is to get VC money to pay for the transfer. So the regulators are quite relaxed with many things, but we need to have a product which is GMP manufactured as an investigational new drug product, and for that you need money, and there is hardly any public funding to finance that, and so you need to convince the VCs.
So funding is a major issue, but then there are quite a few technical issues as well. What are some of the technical challenges we face with EVs compared to MSCs? Because EVs are quite smaller than MSCs.
Yeah, so that's the case. We need to have mainly appropriate analytics, which is not possible yet, or which is maybe a little bit limited. So EVs seem to be very old, so maybe older than cells, and we just have discovered them a couple of decades before, and that is maybe in life sciences. It's either you see it in the light microscopy, in the light microscope, or it's infectious, or until recently we work with homogenates. So that means now we invest a lot of energy in having better analytics, and with the analytics we can evaluate processes. Processes are out in the community for gene therapeutic viruses or other things. We just need to communicate to have the things, and we may also overcome some of the challenges of the MSC field, namely this interdonor variability and aging processes, because we can now use immortalized cell lines which have been raised at the monoclonal level. So since the EVs are below 200 nanometers in size, we can sterilize our products by filtration and get rid of the cells. So with that we have challenges like the analytics, but we have also chances to get a much more scaled, robust production than with the cells.
Yeah, and so talking about analytics, potency assay is a big thing, right? Potency assay on a product where you don't really know what the mechanism of action is sometimes very challenging.
Yes.
Do you think we've learned from the MSC field?
So we have learned from the MSC field that potency testing is quite mandatory. If you discuss with the colleagues from the MSC field and ask them what is one of the largest challenge, it's the potency testing.
Exactly.
Yes, that is what we need to do. We may have also a little advantage compared to cellular products, because if you want to have a cell to interact with another cell type in a dish, you need to consider that the media must be compatible for both cell types. So we extract the EVs up front, and we can just feed them like cytokines to cells in any given media. So it might be an advantage, but of course, thinking about the mechanism of action and to unravel it, it's also a challenge, the same challenge as for MSCs, it will be for the MSC EVs comparably.
So can you tell us what you think is the one thing that can move the EV field forward in the next 5 years?
That's very easy to say. So I believe most of the knowledge is out there. You just need to interconnect people. And for interconnecting people, you need money. So that means we need to convince venture capital providers to invest into the field, but maybe also inherited from the MSC field that there are only 13 market-approved products worldwide. The trust in these new therapeutics is not very high. They are hesitating. Then there is COVID, Ukraine, and now the politics of Trump cutting the budget, that the venture capital funds are in the whole gene and cell therapy very limited. It apparently gets improved, but yes, that's international things where we are also suffering from.
So lastly, you are co-chair for the Exosome Signature Program this Saturday. Can
Yes.
you tell us what we can expect there and what the main themes will be?
Yeah, so this is many of the challenges. We can go much more into detail. So it's the upstream, the downstream process, the analytics that will be discussion part of the first session. Then you mentioned potency testing, mechanism of action, where do we stand? This is the topic of the second session. And then, of course, if you have a product with IND designation and you want to proceed into the clinical trials, you also need to see that you have the right monitoring, the right patient inclusion criteria. And so in the third session, we will have 3 people who collected already experience. So each session will have 3 speakers, each 15 minutes in presentation. And then afterwards, we have the same time, so 45 minutes for the discussion. And as said, one of the knowledge is there. We need money, but we also need to team up and learn to speak one language. And that is one idea of the Signature Series, bringing people together, interconnecting and combining strengths and networks.
Yeah, I've always enjoyed the Signature program. Thank you so much, Bernd, for being here today.
Yes, it's a pleasure
I had a great time talking. Thank you.
Thank you very much.
Here we go. Here. Claire, how are you getting on?
How are you?
We're here with Claire from Nybert. Are you enjoying the conference?
I absolutely am. It's excellent. As usual.
Plenty of good content.
Really good content, really good exhibitions, some good networking.
And it's great to have everybody in Ireland, isn't it?
Amazing, absolutely.
Let's see, can we get as much business, cell and gene therapy business in there?
Absolutely.
Thanks a million, Claire. Enjoy the rest.
See you later.
Take care. Follow me. Pam.
How's it going?
Good.
How's it going?
Good. Enjoying the conference?
I am. I'm having a fab time. It's great to be in Dublin. Never been before. First time in Ireland, so it's been really good.
Yeah.
And just a great conference. One thing I love about ICT is the content. Great content. It's always fun packed.
Brilliant. Any particular content that you enjoyed in there?
Yeah,
What's the kind of key messaging you're seeing coming
I
out?
think there was a really interesting talk yesterday, and it was the focus on patients and donors, and just making sure that the quality of care that they've got is right. Making sure that there's access to patients as well for therapies. I think that's a real issue that we're facing. It'll be interesting to see how we take that away and make changes and how we can get things moving for patients.
Brilliant. Thanks a million, and true. Listen, we'll see you around. Enjoy the rest of the conference. Take care. See you.
Today, I have the honor to talk with Julie Alexson about iPSCs. Hi, Julie, and welcome to the couch.
Hi. Thank you for having me. This is great.
So tell us, why all the hype about iPSCs?
There's just so much potential. So if you think about it, it can be differentiated into any cell. It doesn't have ethical concerns like embryonic stem cells. And I think that there's a lot of opportunities where it's starting to go into clinic. When you look at Japan, they've already got 2 conditional approvals for iPSC. So I think it's starting to really focus on these cells. It's been about 20 years that they've been in research. And I think we saw the same things with MSCs, where it took a while to be able to understand how these can be utilized and how they'll be efficacious. Same that we're seeing now with Mesoblast, with the first commercialized MSC in the US. So it's exciting. I think that we have that same opportunity for iPSCs, besides the opportunities to use them in tissue engineered products.
So a lot of promise, however, they remain very expensive. So can you talk a little bit about the economic hurdles for iPSCs and what makes them so expensive?
Yeah, I think if we think about it, they're in culture for a long time to be able to differentiate. We have to make a different cell from them. We have to reprogram them. I think it's all the reagents and supplies that become very expensive. I think if you look at suppliers that have these, there's an opportunity for us to reduce that cost by working with others or even through partnerships. But I think that the reagents and supplies are probably about 50% of the cost of the product. And then the high cost of labor included with that. So it depends on if you're producing, and not many people look at it as an autologous product. I think that's going to be the most expensive because you're making one at a time. Whereas if we're making an allogeneic and a large bank, and economies of scale allow us to bring that cost down.
Yeah, and then so when we're talking about scaling up, that brings in a lot more challenges, economic and technical. Can you just touch on those challenges?
Yeah, I mean, scaling up, we don't really have anything right now to go from end to end. which I mean all the way from the beginning, the cell isolation, the cloning, the selecting the cell, and then getting it all the way to fill finish for the master cell bank, working cell bank, and possibly even differentiation. So I think once we get more automation, and there's definitely automation, but it just doesn't cover every step. We don't have a connection all the way through. And I think once that happens and we're working with economies of scale, I think there's an opportunity to start bringing that down. There's going to be more interest. Hopefully people start to commercialize these therapies, and I'm hoping that we'll follow and see the cost reduced.
So what is the biggest innovation that you're excited about in this field to address some of those challenges?
You know, as I said, the automation. I think public-private partnerships are going to be key, because someone has to fund these unless we have investors that come in and want to fund everything. I think that there's large opportunities right now for large grants such as ARPA-H in the US, where these things are being funded possibly at the $30 million point, which has been incredible to be able to move it along. So I think that's one of the ways that we can really accelerate the technology. We have recently received the print grant from ARPA-H, which we're working with Carnegie Mellon on that, and allows us to move forward with a tissue-engineered liver that would be with immune-cloaked cells and differentiated. So I think these types of innovations, as we're using them for things that have never been done before, I think this really gives us an opportunity.
Yeah, and the topic of funding has come up almost every day this week. So it seems like that is really a major barrier for many therapies, but especially when it comes to iPSCs.
Definitely.
So I also wanted to ask you to talk about the iPSC committee that you founded. Can you give us a little bit of insight on how you hope to move the field forward through that committee?
Yeah, thanks for asking that. I think, ISCT has been a part of my life for a long time. I'm one of the founding members of ISCT, where it was a small group of people, maybe 100 people in a room, which really started out with bone marrow transplant and then moved to what it is today, which is incredible. We have a work group that I founded called the Emerging Regenerative Medicine, IPSC, and bringing leaders together that are under process development and manufacturing. We are very productive. We've written 2 papers on scale up. In fact, a 2025 paper just received the award for the most downloads of that paper for scaling up. So it really gives you step-by-step on how to scale up. We're super excited about that. We're working on a gene editing paper for allogeneic cell sources. We have a roundtable every year. So it's a great opportunity for people to get involved. So I hope that anybody that's listening to this, please reach out and join us, because I think we're very productive. If you have an interest in iPSCs, there's a lot to learn. And also to join ISCT in general. I mean, you yourself see what advantages there are to being a part of this community and networking.
Yeah.
It's been amazing for me over 30 some years.
Yeah, it's truly like a family, and a very collaborative family. So I've been very happy attending these meetings. Julie, thank you so much for coming to talk with us today and for giving us your time.
Thank you.
Thank
Really
you.
appreciate it.
I have the honor to speak to Bruce Levine about a hot topic in the field, which is in vivo CARs. Hi, Bruce, and welcome to ISCT TV.
Great to be with you.
So, like I said, it's a hot topic, in vivo CAR Ts. Why all the excitement?
Yeah, it sure is. And I think the excitement is the potential availability to many more patients than ex vivo CAR T. Ex vivo CAR T requires taking the cells out, bringing them to a laboratory, the manufacturing, the testing, and then returning to the patient. The average vein-to-vein time is 3 weeks. Often it's longer than that. With in vivo CAR T, this could be in the freezer, available to a patient upon determining eligibility.
Yeah, so it really shortens that timeline.
Yeah.
And do you think this therapy is going to go beyond oncology faster than, say, ex vivo CAR?
Well, there are 2 potential applications for in vivo CAR T. One would be in oncology, and there are companies that are developing in vivo CAR T in oncology and testing in clinical trials. And they've started in the blood cancers, lymphoma and myeloma primarily. But again, the attractiveness is the immediate availability on determining eligibility. Contrast that to ex vivo CAR T, and there are patients that are not served because of the manufacturing slots and also the cost. The other application for in vivo CAR T is in autoimmunity, where in oncology, the approach is to use a viral vector. In autoimmunity, the approach is to use lipid nanoparticles encapsulating mRNA. That's a temporary CAR, right? But in autoimmunity, you don't need a permanent CAR. You actually may not need to kill every B cell, but just enough to reset the immune system. There's just a handful of patients that have been treated in oncology and in autoimmunity, but the early results that we've seen are really striking.
So talking about those early results, what are we seeing in terms of safety and efficacy?
Yeah, so safety, it's hard to tell with small numbers. There's been a report where there's signs of efficacy, but also there are concerns about the safety with the viral vector in vivo CAR T. In the lipid nanoparticle, again, we're seeing some early results on B-cell depletion. Maybe less of a concern with efficacy because the gene is not integrating into the genome, but still we're concerned in both cases with potential immunogenicity. I think with the viral, it's probably a one-shot deal that you give a viral vector and you may not be able to give a second one because of the immunogenicity. The lipid nanoparticles, again, early days, we don't know how many injections you can give.
So how do we know, what are the concerns regarding to where are these CARs going? Are they going to the right place when we're talking in vivo CAR?
Yeah, that also gets to the design of the in vivo CAR T. With the lentiviral vector, they can be generated with a targeting, an antibody targeted against a marker on the cell surface. The lipid nanoparticles come with and without, but lipid nanoparticles with targeting can find T cells. The question always is, where else could they be going? But the targeting and the design of the LNP and the design of the viral vector goes a long way towards demonstrating that in animal models, and we need to validate that in human trials.
Great. And so tell us, what do you see this field doing in the next, say, 5 to 10 years in both oncology and beyond?
I think we're going to see the emergence of in vivo CAR T. I can't predict yet whether it will be a viral vector or LNP in autoimmunity or in oncology, but with the potential to treat many, many more patients around the world and in a much more cost-effective fashion, there definitely is incentive to develop this.
And bringing hope to so many more patients.
Yes.
Thank you so much for being with us today. It was an honor to talk with you.
Yep, thank you very much.
Thank you.